Thanks, everybody. Happy Friday. Happy Groundhog Day, actually. Today, and I wanted to say thanks for everybody for coming. Today, we're going to talk about chikungunya virus-specific CD4 T cells and how they're associated with chronic viral arthritic disease in humans. And Daniela Weiskopf from the local Institute of Immunology and Center for Vaccine Innovation is going to be giving us her talk. Next slide.
Mm-hmm.
Just a few things today about today's speaker. She's a research assistant professor at LJI in the Center for Sex-Based Differences in the Immune System and the Center for Vaccine Innovation. Prior to running her own lab, she was also an instructor and a postdoc at LJI and has a PhD in immunology from Innsbruck Medical University and a master's in microbiology and immunology, also from Innsbruck. She's devoted her career to understanding T cell responses in infectious diseases and human health, and she's going to tell us about that today in relation to chikungunya, which causes all sorts of problems, especially achy joints. So I will turn it over.
Great. Thank you so much, Davey. Daniela, I'll pause the share and you can start whenever you're ready, and then we'll have a Q&A afterwards, everyone. All right. Thanks so much.
So what do? I need to share it from my screen now?
Yeah, share from your screen now, yeah.
For some reason, I thought like that my slides will be behind these slides, but they're not, yeah?
I do have them. I can pull them up. You'll just have to tell me when to advance. So just let me know if you want to do that.
Oh, okay, yeah, let's do that because I for some reason I thought they are in the slide deck. I'm sorry.
No worries. No,
mistake. Okay.
1 second. Presentation mode. Welcome. Sorry.
Yeah, so while Force is helping me out of my my misery here, I work at the La Jolla Institute of Immunology. I have always been interested in emerging viruses. I worked a long time, more than 10 years, on dengue virus. Zika, when Zika emerged 2015, 16 in Latin America, then I've been working on that. I have also been working on yellow fever, vaccine-induced responses, and another mosquito-transmitted virus, which I'm telling about today, is chikungunya. Chikungunya virus is transmitted by mosquitoes, the same mosquitoes as that transmit the other flaviviruses. This is not my slides, right?
No, this is the slides that you shared with me, yeah.
No. Okay.
1 second, everyone.
Fine. All right. I will share my screen now.
Great. Thank you, Daniela.
All right. All right, here we go. So, as I said, what I'm showing today is like a part of my research on chikungunya virus. Chikungunya, in case you don't know where the name is coming from, it's refers to like the African word, the people that are bent, because that is basically like how the disease manifestation looks like when people get infected with this. If you're interested in the virological background, chikungunya is a positive sense single-stranded RNA virus. It is part of the tocaviridae, and as I just mentioned, it is part of the mosquito-transmitted viruses, so it's an arbovirus, which is the overlapping family of flaviviruses and alphaviruses. So it basically consists of 2 open reading frame. It has 4 non-structural proteins and 5 structural proteins. And these are basically spanning like different size of amino acids. and it looks very much similar to like other alpha viruses that have been that are known. So chikungunya is transmitted and distributed globally. So it basically goes between sylvatic cycle, non-human primates and mosquitoes, but then it can always, and it does very often, switch to an urban cycle where like the human is the intermediate host between the mosquitoes. So once you're infected, you can be bitten by a mosquito that can then transmit the virus to somebody else. So as you can see here, this really is a global transmission. So in all of these tropic and subtropical areas, they can be transmitted, and that is from Latin America, Africa, and also Southeast Asia. There's different lineages that are known for chikungunya viruses, and it depends on the outbreak which one of these lineages is spreading. So why I'm very excited to show this in terms of a Pandemic Preparedness Institute seminars, because like this transmission is very much going on right now. So up to like December 2023, so last year, there were over 500, close to 500,000 cases and over 360,000 deaths reported. Right now, it's a majority like spreading in Latin America, but there's also cases in Africa and Asia have been distributed. So, and I've just been like, you know, looking at the numbers from this year, 2024, and it doesn't look like it's calming down. So it's even like moving closer to cities, and as soon as you have a lot of hosts in between, you have really closing the cycle. So we're probably not gonna see like calm down of situation like anytime soon. Is there anything that you can do against chikungunya? So just right now, a couple months ago, there has been one vaccine licensed against chikungunya, which has been like produced by Valneva. It has been licensed by the FDA, has not been distributed, is not available right now, but so there is some vaccine that is will be available soon. So what is so interesting about the chikungunya is really the pathology. So if you get infected, it takes about 3 days to symptom onset. So almost everybody gets symptoms in the acute phase that is signified by fever, headache, retroorbital pain, rash. The majority of people in subacute phase can clear the virus and also releases all of the symptoms. But what is peculiar about chikungunya is that there is a portion of people, 30 to 60%, and that depends on the viral strain that is infected. Some of them are more prone to induce that, some of them less. So I would say 30 to 60% of these infected individuals never get rid of the symptoms. So they enter a chronic phase that is signified by myalgia, and the hallmark of this chronic phase is arthralgia. So they have joint pain and swelling, and it's referred to as chronic inflammatory rheumatism. So that's something that is particular about this virus, because typically, like we think about these arboviruses that they are acute, you clear them and you're good. But so in this case, there's a big portion of people that go out of the acute phase and then enter a chronic phase. So what is interesting is where does the virus go if you still have symptoms and you can't clear the symptoms? So is the virus, but does that mean the virus is still there? So, and that is something that has been looked into, not by us, but by others. Eileen Cheng from George Washington University, they have been looking into synovial fluids, because that's where like all the symptoms like occur in a lot of people, and they have never seen virus. So there's for some reason there is like symptoms still around, but the virus is gone. So it's unclear like why that is. So overall, as I just pointed out, this has been reaffirmed as rheumatoid inflammatory arthritis. The symptoms are similar, so they both affect small joints such as fingers and wrists, and also large joints such as knees and elbows are infected. Joint edema is present, and fatigue, myalgia, insomnia, and depression can also be present in both of these disease manifestations. There's not really biomarkers available. There has been cytokine profile established, What we do know is that it primarily affects middle-aged women. So 60 to 80% is women. Another like hallmark of typical like autoimmune disease, where more than not are affected, women are more affected than not. You would think that you could maybe use drugs that are like, we know, like work for anti-rheumatic drugs, and they have been tried and they are effective in some cases, but certainly it's not like one fits all. So there's something that might be different than that in the disease pathogenesis from compared to outbreak infection. So what do we know about the immunity against chikungunya? So not so much so far. So this is the only publication that has ever looked at T-cell responses to chikungunya in humans, and that is from the La Reunion outbreak. And they have looked 12 to 24 months post-infection, and they picked 3 out of the 10 chikungunya proteins, and looked if there is a recognition of this, so if interferon gamma is produced by that, and then also is there a difference in people that can still show symptoms or have cleared the virus or the symptoms. So you can see they didn't see a difference in the 3 proteins they looked into that. So when I started looking into more detail, I realized this is kind of the only thing that we know about chikungunya. So we were wondering, well, so if we look into this, how far out can we see like T-cell in the periphery, like, you know, how many years post-infection? What are the proteins that are recognized? Because like this publication showed the response to 3 proteins, but the chikungunya has 10. So what is the immunodominance against this virus in the terms of T-cells? And then what are the T-cells actually look like, you know, in people that have chronic symptoms compared to those that had recovered? So that's all unanswered questions that the field really does not know so far. And following this according to the phenotype, so we also don't know what is the functional profile of these cells. So this is the cohort characteristics that we have been recruiting. So these samples come from people that have been infected in the Northern Colombian era, Barranquilla. So this is basically people that have been infected in the last big outbreak in Colombia in 2015, 2016. And we have been collecting samples from these donors years after they have been infected. So overall, we have been collecting 31 seropositive donors. So these people have had symptoms when they were acutely infected. They do show seropositivity against this virus. And we compared it to chikungunya seronegative donors from the exact same cohort. So just we can like, you know, set our assays to a negative background. So overall, I can already tell you that also our cohort contains mostly females, so 87% of females, and the seronegative cohort is like more balanced between male and female. On average, the cohort is 42 years. Our control cohort is like 38, and then they have been recruited on average 6 years after diagnosis. So that means they all have had a positive diagnosis against chikungunya, and we have been following up like 6 years after. And this is basically to remind you again the structure of the virus. The way we doing T-cell assays is something that referred to as peptide megapools. And that has the advantage that you can make overlapping peptide sequences spanning the entire like proteome of the virus. So you basically start from here and you go there make 15 MERs and always shifting them by 10 amino acids. So you basically have a complete, fully like comprehensive like screen of the viral sequence. And that is of advantage if you want to study like a population that where you do not know the genetic background. So if you do not know what HLA phenotype these people do express, if you do not know what is an HLA phenotype that you want to look for a certain virus, then you can use this assay because it's very agnostic. It will like pick up all of the T-cell responses from the entire proteome, regardless of like the genetic background of the host. The assay that we're using is something that referred to as AIM assay. It's an activation-induced marker, And it basically works also very agnostic. So it works. It will like identify antigen-specific T cells based on the activation of in activation-induced markers, and that is irrespective if these T cells can produce cytokines or cannot produce cytokines. And that is another a very agnostic approach. When you do not know what cytokines do you expect to be produced, for certain viruses, if a new novel virus is emerging, we many times do not know. This is going to be a classic virus that makes a different gamma between alpha 9 interleukin 2. So you use this assay because this will pick up like all of the antigen-specific cells regardless of the cytokine they produce. And also it will pick up all antigen-specific T cells regardless if they even do make cytokines. For example, T follicular helper cells, we know in the periphery do not make a lot of cytokines. So this assay will pick up all of them. So two very agnostic approaches if you don't know what you're doing and don't know what you expect. So you can pick the megapole approach with like overlapping peptides spanning the entire thing, so you do not miss anything. And you can use this assay if you do not know what cytokine you need to like look for. And this is basically what we have been doing. And this is going right into like our results. So we have first looked into what are the CD8 T cell targets. And we were quite surprised to see that CD8 T cell responses were not detected. So you can see there's very, very little like people that have responded to any of these polls with CD8 T cell response. But this is like surprisingly low and not what we would expect from a viral infection. And you can see here as a control, we always run cytomegalovirus pools. So that is just a representative for people that have been infected. So it's not a problem of these people because they're very well able to make CD8 T cell responses against other viruses, but not chikungunya. And of course, I told you like that we're gonna always like benchmark our assay against seronegatives, and you can see like if you have never seen the virus, so you will also not respond to our peptide pools. So when we looked at the CD4 T cell responses, the picture looked quite different. So again, in a seronegative, we do not see a substantial response, but we do see quite a substantial response and strong response against almost all of these proteins from the CD4 T cells. So there is antigen-specific CD4 T cells in these donors that strongly recognize a big variety of these proteins. And they look very comparable to like, you know, CD4 T cell responses that you see from a chronic virus such as cytomegalovirus or a vaccine antigen that people get like, you know, every 10 years, tetanus. So that was surprising because this is not what we would expect from a classic viral infection. So we were thinking, what do these CD4 T cells still see if the antigen is not around? Because I just showed you in our introduction slide that nobody has ever found the virus in these patients once they cleared the symptoms. So we looked into an assay that can pick up like recently activated T cells. And this is basically done this for chikungunya specific CD4 T cells compared to cytomegalovirus. Because cytomegalovirus is something that like most of us have like been infected with and constantly reactivated every couple of years. So this is what we compared it to, and you can see it does not look like some viral profile where you have recently seen the antigen. It looks much more like a case, in this case, a tetanus toxoid, which people do get from the vaccine, which has very similar activation levels than chikungunya. So this does not look like the CD4 T cells have been recently activated, yet they're still around 7 years after. So to answer the first part, yes, we can detect CD4 T cell responses, mostly CD4, but not CD8, 6 years post-infection. So I've already shown you that there's a wide area of proteins recognized. So, but what exactly? So we looked into this and analyzed this based on what's the percentage of response, which is shown here in the black bars. What's the percentage of responders? So how many of our donors have responded? And is the response, the immunodominance, as a function of like the the size of the protein. So that means the larger the protein is, the more people responded. So that's basically what we looked at. And the answer is like the size does not give you any inclination of what is like immunodominant recognized protein. Because you can see here, this is a fairly large protein. Is recognized by less than 20% of the donors, but if it's recognized, the response is fairly like strong, which you can see here in the blue. But we also have proteins which are like really large. The largest protein, non-structural protein 4, which is not recognized and also not by a high frequency of responders. We also have the case where the protein is not that large, but is recognized by a high percentage of responders and also has a significant negative response. So that's interesting because now that gives you like an exact profile of what are the proteins that are recognized in chikungunya. And you can see here E1, one of the structural proteins, is the most dominantly recognized, shortly followed by non-structural protein 1. And then it goes down some of the other proteins. So that is just giving you an idea of what is the immunodominance of this viral response. And that's what I've just been telling you. So we were then interested in, is there a difference in the case of people that have been infected and have cleared the symptoms, or if there's people that have been infected but still have symptoms 6-7 years after infection. So we were interested in like, do the T cells look any different? And so we split our cohort in people that have symptomatic still infection or are recovered. And you can see here, they are basically diagnosed with arthralgia 6-7 years after they have been infected, and we compared it to people that have cleared the symptoms. If you wonder if we could already tell the time of infection, but the symptoms they displayed, if you could already tell then, you probably will get chronic infection, you will not. The answer is no. They look completely the same at the time when they were acutely infected, but they look very different now, 6-7 years afterwards. And you can see overall, of course, the ones that are infected are mostly female, but that's because our entire cohort is mostly female. And on average, they are like 6 to 7 years after they have been infected. So is there a difference in antibodies? So that was the first question we asked. So if you have been infected and you have recovered, do you have like a higher amount of antibodies or lower amount of antibodies? And the answer is no. It looks exactly the same. You cannot tell based on the levels of antibody titers who belongs to what group. What you can tell though who belongs to what group is like if you look at the CD4 T cell responses, and I told you like it's about CD4 because this virus does not elicit CD8 T cell responses, and there's a highly significant difference in people that still have symptoms compared to like people that have recovered. And when we look into each of the individual proteins, like where does this difference come from? It's really true for almost all of the proteins, as you can see here, but significantly higher recognized in symptomatic patients is NSP1, one part of the NSP2 protein, and the E2 protein. So we can significantly show that there is higher CD4 T cell response, chikungunya specific CD4 T cell response, and that tracks to like some of these proteins that I just pointed out. So do they overall recognize different proteins? Because I showed you here that like, also the red ones do see a lot of variety of proteins, but the answer is the magnitude is very different. As I just showed you, there's a lot higher CD4 T cell responses compared to recovered, but the immunodominance is fairly similar. So probably if we would have looked like much closer to infection, they would have had the exactly same response. And that kind of goes together with what we see from this very first study from Larion that looked into like 12 to 14 months after infection, where they did not see a difference in these 3 proteins. So what is different then in these T cells? So is it just more of them, or is also the phenotype different? So that's what we looked into next. And you can see, if you look at the memory phenotype, so what are the subsets of memory that is present in these different donor groups, you can see there's no difference. So most of them still have central memory cells, effector memory cells, and there's some temporal cells. And there's no difference in the group that still has symptoms or the group that is recovered. We then looked at the T helper subset. So that means, is the cell more like a Th1, a Th17, or a Th2 cell cytokine? And we did start seeing differences there. So interestingly, the Th1, which is typically associated with viral clearance or defense against the viral viruses, is a lot stronger, significantly stronger in people that are recovered compared to the ones that have symptoms. So there's no significant difference, but if you're wondering where this difference comes from, because if you have by definition lower Th1, you must be higher in something, and it looks like there's a trend to higher Th17 cells. And interestingly, like Th2 is completely the same between these two groups. So to summarize that, yes, there is CD4 T cells that are frequently more present in symptomatic donors, and they start pointing towards different phenotypes in terms of the helper subsets. So does this relate into different functional profile? This is the last question that we were interested in. And the way we're looking into this is like, what cytokines can they produce in response to chikungunya antigens? And how many of these cytokines they can induce, because it's typically understood that if you have polyfunctional response, that means they would be like in this blue section here, they can produce two or three more cytokines. The same cell can induce that. That's typically associated with protection from like infection. So, but what we do see here is like 71% of our antigen-specific T cells only make one cytokine. That is surprising because like that's not what we would expect from a typical antigen viral response. So then when we looked into like what is the cytokine they produce, this was our next surprise, because like there's a significantly production of TNF alpha and not so much of interferon gamma, which is also not what we would expect from a viral infection, where we would expect a lot more interferon gamma specific responses. So we compared this in the exact same donors. This is paired samples from the same donors. If they either respond to chikungunya or cytomegalovirus, which I've been showing you is like always used as a control. And there is significantly like less interferon gamma produced in this donor against chikungunya, but not cytomegalovirus. And it's the other way around for TNF alpha, which is significantly higher for chikungunya specific T cells and lower for cytomegalovirus. Interestingly, they also make significantly less interleukin-2. So this could go well with like an observation that has also not been made by us, but by Eileen Cheng from George Washington University, is that the T regulatory cells might be different in these people. And that would go well with this because they do make significantly less interleukin-2 compared to like a classic chronic infection. There's no difference in IL-10, and there's also no difference in any of the other cytokines that we have been looked, IL-17, IL-4, but we haven't seen the response against that. So to conclude that, we do see individuals with chronic symptoms that have a higher proportion of chikungunya-specific CD4 T cells, and we showed that they are specifically higher in NSP1, NSP2, and E2 proteins. We do show that they have a lower Th1 phenotype, but are enriched in Th17 phenotype, and they are primarily only able to make one cytokine, so they're monofunctional, and the one cytokine they know how to produce is TNF alpha. So that looks awfully closely to when you look at rheumatoid arthritis CD4 specific T cells. So it does look like there is certainly a significant difference in the CD4 T cell profile in people that have still a chronic infection versus the ones that have been cleared. And it does suggest that the CD4 T cells that we see potentially play a pathogenic role in this chronic chikungunya disease, because like we do see them significantly enriched in people who still have symptoms. So that's what I wanted to share with you. And these are the people here marked in bold that have been involved into this study. So almost everybody from my lab. And we had our collaborators from Universidad del Norte in Barranquilla, Colombia, who helped us to collect these samples. And UNC, Mark Heise and Prem Lakshmane have been instrumental to help us to like measure the antibodies against this virus. And with that, I'm happy to take any questions.
That was great. Anybody have any other questions?
There's a few in the chat,
Chat,
actually.
yeah.
So I know Steve had asked, and this was maybe earlier on, about the seronegative cohort members, if they had a chikungunya diagnosis. They did not. Okay.
No.
Okay. And then also, Vivian had asked, did you look at the T regulation in recovered versus chronic?
Yeah, no, I haven't, but that's like, that awfully looks like where this is pointing to, because there's like significantly less interleukin 2. So that is the next logical step to look into it. Yes, I have not yet, but yes, I agree with that would be the right way to do it.
Okay. Anyone else have any further questions about today's talk?
Yeah, what are you most excited about going forward?
Yeah, well, first, what I am most excited about is like to see why are these T cells around? They must see some antigen. So I don't know. If you ask everybody, there's no virus like in these people, but so I don't know. So UCSC actually does collect the tissue samples from the synovial joints. So we can see if there's CD4 T cells in there and not virus. That would be interesting. And then, I mean, yes, what they see, I don't know.
And then any lingering viral pieces, it doesn't have to be viable.
Yeah,
virus.
exactly. Yes, exactly.
Okay. Any other thoughts, questions?
Sounds good. Davey, I'm just going to present the next speakers,
Yep.
and we'll
Go for it.
Yeah. All right, everyone. So again, on behalf of the Prepare Institute, thank you all for taking part today and hearing Dr. Weiskopf's talk. It was excellent. And we just want to let you know that we have good speakers lined up for next month. We'll have Drs. Ratna Shlar and Sandra Leibel, who are actually recipients of a co-developmental grant that Prepare had with the Human Milk Institute, HMI. So we're still working on the details as to when that will happen, but please do show up next month. We really appreciate you all being here. So with that said, thanks so much for being here again, and have a lovely weekend, and we hope to hear from you soon. Thank you so much, Daniela.
Bye-bye. Thank you.
Thanks,
Thank
Daniela.
you.
Take care, everyone. Bye-bye.